I'm Dr. Khokon Kumar Dutta. My Christian name is Dr. David Moon. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Research Advancements Toward Pancreatic Cancer Treatment Targeting microRNA-34a. Khokon Kumar Dutta et al. For the first time succeeded to detect the presence of mature microRNA-34a in both exocrine and endocrine cells of the rat pancreas. We used in situ detection approach to localize mature microRNA-34a in the pancreas. We used highly specific mercury microRNA detection probe of our Danish company exocrine. Internationally, this was the very first time detection of the presence and involvement of microRNA-34a in the pancreatic tissue of any living organism. We also investigated the differences in expression between normal and cancerous tissues. We were lucky to get some samples of human pancreatic cancers from South Korea. Actually, we investigated 10 different samples of human pancreatic cancers. All these samples were collected from pancreatic adenocarcinoma type. We found a mixed type of expression pattern of microRNA-34a in the pancreatic cancers. We found high level of microRNA-34a presence in only 10% of the samples. 30% of the samples showed very low level of presence of microRNA-34a. The rest 60% of the samples showed the moderate level of presence of microRNA-34a. We published our findings in September 2007 in the Journal of Cancer Science entitled Association of microRNA-34a overexpression with proliferation is cell type dependent. All the research works were done in the School of Medicine, Kyoto University, Japan. Shung Sheng Shang et al. Published an article in June 2007 in the Journal of Molecular Cell entitled Transactivation of microRNA-34a by p53 broadly influences gene expression and promotes apoptosis. The group used cultured cells of pancreatic origin for their experiments. We were not informed about their works and findings as our first documented submission was made before their article got published online. Here I would like to mention randomly some of the important works rating the therapeutic potentials of targeting microRNA-34a in pancreatic cancers. I would like to be sorry for not mentioning all the pioneering works that made significant contributions in exploring the critical roles of microRNA-34a in pancreatic cancer at least. Kewing G. et al. Published an article in August 2009 in the Journal of PLOS ONE entitled microRNA MID34 inhibits human pancreatic cancer tumor initiating cells. CD44 positive and CD133 positive cells behave like cancer stem cells or tumor initiating cells. The group found low level of presence of microRNA-34a, microRNA-34b, and microRNA-34c in CD44 positive and CD133 positive MAIA-PACA2 cells. High presence of BCL2 and NOS1 was found in CD44 positive and CD133 positive MAIA-PACA2 cells. The count of these special type of cancer cells were only 1 to 2% of the total cell populations. microRNA-34a restoration resulted about 87% reduction in the number of CD44 positive and CD133 positive MAIA-PACA2 cells, accompanied by significant inhibition of tumor sphere growth in vitro as well as tumor formation in vivo. microRNA-34a restoration inhibited colonogenic cell growth and invasion, induced increased apoptosis, and G1 and G22M arrest in cell cycle. The group also found that restoration of microRNA-34a makes pancreatic cancer cells 2 to 3 times more sensitive to both chemotherapy and radiation therapy. Yan Chen et al. Published an article in November 2019 in the Journal of Experimental Clinical Cancer Research entitled Circular H2L Promotes Tumor Progression by Sponging MAIA-34a to Regulate NOS1 in Pancreatic Ductal Adenocarcinoma. A circular RNA H2L highly expressed in pancreatic cancer cells and involved in the promotion of tumor invasion, proliferation, and angiogenesis. Circular H2L regulates microRNA-34a, thus become able to regulate NOS1. Circular H2L served as a microRNA response for MAIA-34a and promoted tumor progression in vivo. The group also analyzed circular H2L in clinical samples and found a correlation with lymphatic invasion and TNM stage. Anna Sastre et al. Published an article in June 2020 in the Journal of Nature Scientific Reports entitled MAIA-34a Constrains Pancreatic Carcinogenesis. The group found that MAIA-34a knockout mice, or in the absence of MAIA-34a mice, rapidly develop the lesions for the pancreatic cancers at an early stage. A combination of the absence of microRNA-34a and the presence of constitutively active oncogene KRAS make pancreatic cancers aggressive at a very early stage. In this particular combination, invasive pancreatic cancers developed in 6 months of age in mice. The group also reported that microRNA-34a ablation leads a cell autonomous activation of the inflammatory pathways which cover at least a third of the contribution of microRNA-34a in pancreatic cancers. The anti-inflammatory roles of microRNA-34a is executed by the suppression of TNF-alpha and interleukin-6. Zhou et al. Published an article in August 2020 in the Journal of Drug Design, Development and Therapy entitled Exosomes-Coded microRNA-34a Displays Potent Anti-Tumor Activity in Pancreatic Cancer, both in vitro and in vivo. Exosomes are nano-sized vesicles with a diameter of 30 to 150 nanometer and found in many body fluids, including blood, cerebrospinal fluid, saliva, urine, and milk. The group purified exosomes from cultured HEC293 cells and prepared exosomes coded microRNA-34a, exomit-34a, using ultrasound approach. The group confirmed the high level of transfection efficiency, 86% for exomit-34a, in PANC28 and MAIA-PACA2 cells. They also confirmed the in vivo drug delivery efficiency of exomit-34a, including a positive control 5-fluorouracil 5-APU loaded exosomes in their experiments. In case of in vitro experiments, exomit-34a induced 79% and 67% growth inhibition in PANC28 and MAIA-PACA2 cells respectively. The group found that exomit-34a was able to inhibit cancer cells growth and to induce cancer cell death in apoptotic pathway by targeting BCL2. They prepared mice with pancreatic tumors of at least about 80 cubic millimeter for in vivo experiments with exomit-34a. They used xenograft nude mice model and subcutaneously injected PAC28 cells for the tumor development. Exomit-34a IV was administered when the tumor grew at least about 80 cubic millimeters and was checked other day for 21 days. At day 21, after administering exomit-34a, the inhibition rate for tumor growth was found to be about 86%. Hedas Gibari et al. Published an article in January 2018 in the Journal of Nature Communications entitled Amphiphilic Nanocarrier Induced Modulation of PLK1 and MIT-34a leads to improved therapeutic response in pancreatic cancer. The group found that combinatory treatment of microRNA-34a and siRNA for PLK1 together increase the survival length in pancreatic cancers. They also found that microRNA-34a and PLK1 reduce the level of oncogene MYC in pancreatic cancers. The group developed and used biodegradable amphiphilic polyglutamate amine polymeric nanocarrier APA for the effective delivery of both microRNA and siRNA in both in vitro and in vivo experimental systems. Xiao Fei Jin et al. Published an article in April 2019 in the Journal of ACS Applied Materials and Interfaces entitled Ross Responsive Polymeric Mycelis for Triggered Simultaneous Delivery of PLK1 Inhibitor and microRNA-34a and Effective Synergistic Therapy in Pancreatic Cancer. microRNA-34a is now a well-known suppressor gene for the pancreatic cancer, and PLK1 is closely associated with short survival rates of pancreatic cancer patients. The group used polyethylene glycol, polyaspartamidoethyl p-boronobenzyl diethyl ammonium bromide to deliver microRNA-34a and polo-like kinase 1 PLK1 for both in vitro and in vivo experiments. The polymer could self-assemble into micelles of approximately 100 nanometer with 10% drug loading of bolazertib and form a complex with microRNA-34 at the NP ratio of 18 and higher. The group found superior anti-proliferative effects, enhanced G2 to M phase arrest, and suppressed colony formation using a combinatory treatments of cancer cells and tumors with microRNA-34a and PLK1 together. They found a negligible level of unwanted toxicity in this treatment of pancreatic cancers. Mano Nanamoni et al. Published an article in January 2021 in the Journal of Oncology Letter entitled Circular Dumbbell microRNA-34a 3p and 5p suppresses pancreatic tumor cell induced angiogenesis and activates macrophages. Angiogenesis is a very critical factor for the tumor genesis and cancer. A tumor cannot grow more than 1 to 2 cubic millimeter if it fails to collaborate successfully with the angiogenic process for the increased supply of oxygen, nutrients, and others through the newly formed blood vessels. Many scientists are trying to limit the angiogenic process to fight against the tumor and cancers. This group targeted the cancer-related angiogenic process for their strategy. They made circular dumbbell form of microRNA-34a 3p and 5p DB34a to study the effects on angiogenesis and on the immune responses. The group used tube formation assay to evaluate the effects of DB34a on angiogenesis in vitro and in vivo. DB34a transfected cells showed significant reduction in tube formation compared with the cells transfected with scrambled dbRNA. They used zebrafish angiogenesis model for the in vivo experiments. The results of the in vitro experiments were successfully validated in zebrafish. The group also found that conditioned media collected from DB34a transfected PANC1 and MAIA-PACA2 cells was able to induce inflammatory responses in macrophages. Interleukin 13, CCL5, and CXC motive chemokine ligand 12 alpha, CXCL12 alpha, among other inflammatory response genes. Jia Shun et al. Published an article in April 2018 in the Journal of Oncology Reports entitled Long Non-Coding RNA GIST Exert Oncogenic Functions in Pancreatic Cancer via microRNA-34a 5p. An upregulated GIST was found in the pancreatic cancer tissues. Elevated presence of GIST significantly promoted the proliferation, migration, and invasion and suppressed apoptosis of BXPC3 cells. On the other hand, knockdown of GIST significantly inhibited the proliferation, migration, and invasion and accelerated cell apoptosis of PANC1 cancer cells. The group found reduced presence of microRNA-34a in the pancreatic cancer cells. microRNA-34a was the target of GIST in pancreatic cancers. The level of microRNA-34a was negatively correlated with GIST. microRNA-34a was able to rescue pancreatic cancers from GIST-induced malignant behaviors. Hung Wei Wang et al. Published an article in December 2020 in the Journal of Bioengineered entitled Propofol inhibits migration and induces apoptosis of pancreatic cancer's PANC1 cells through microRNA-34a mediated E-cadherin and log285194 signals. Propofol is well known to exhibit potent anti-tumor activity in pancreatic cancer cells in vitro and in vivo. The group tried to explore the anti-tumor mechanisms of propofol in vitro PANC1 cells. Propofol induced the elevated presence of microRNA-34a to induce apoptotic death in cancer cells. microRNA-34a pushed cancer cells to death by upregulating log285194. microRNA-34a also induced upregulated expression of E-cadherin, which inhibited the migration of cancer cells. Elizabeth Landau made a report on CNN on November 9, 2020 entitled Why Pancreatic Cancer is So Deadly. According to the experts, 95% of the pancreatic cancer patients die from it. Even in the early stage, pancreatic cancer is lethal. In 2018, pancreatic cancer was the third leading cause of death from cancers in the United States of America. An association with race was also reported. African Americans are more likely to develop pancreatic cancers than the white. According to Cancer Research UK, 24.8% of the males survive pancreatic cancer for at least 1 year. This falls to 6.5% surviving for 5 years or more, as shown by age standardized net survival for patients diagnosed with pancreatic cancer. Survival for females at 1 year is 26.2% and falls to 8.1% surviving for at least 5 years. Survival for females is higher than for males at 1 year and higher than for at 5 years. The summary was made on the data in between 2013 to 2017 in England. According to the World Health Organization's data published in 2018, pancreas cancer deaths in Bangladesh reached 930 or 0.12% of total deaths. Research on the exploration of the roles of microRNA-34a, an efficient and safe delivery system, may help to reduce the huge burden of pancreatic cancers worldwide, including Bangladesh. Only the information known to me was discussed above. I may miss some critical words to include. I welcome your criticism for the moderation and upgradation. Finally, I would like to wish happy, prosperous, and joyful days for you, everybody. Thank you.